Welcome to the module entitled What is Genome Sciences? My name is Suzanne Haga. I am an associate professor in the Duke Center for Genomic and Computational Biology at Duke University. This module was developed by myself and Dr. Greg Ray of GCB. The objective of this module is to provide a brief overview of genome sciences, tools and technologies, and applications. First, what is a genome? A genome is the entire DNA content that is present within a cell or organism. So what is genome sciences? It is the field of study on the development and application of genomic, transcriptomic, proteomic, metabolomic, and computational analytical tools to address a variety of basic and applied science topics. In some studies, one or more technologies can be used per sample or specimen. What is the difference between genetics and genome sciences? Genomics typically refers to the use of technologies to analyze all of a certain type of molecule in a tissue or cell. For example, a genomic study can take a snapshot of a certain type of molecule, like the entire genome, or it can involve analysis of hundreds to thousands of samples, thus generating very large data sets. In contrast, genetics typically uses technologies to analyze one or more genes. It usually focuses on suspected genes or genetic variants of interest or specific region. In medical genetics, family-based studies are often performed compared to the population-based studies performed in genomic sciences. Several technologies have been developed to comprehensively analyze various molecules rapidly and accurately. For DNA, whole genome sequencing, whole exome sequencing, and microarrays may be used to analyze the genome. For RNA, RNA sequencing, or RNA-seq, single-cell RNA-seq, and microarrays may be used to analyze the transcriptome. For proteins and metabolites, mass spectrometry and nuclear magnetic resonance may be used to analyze the proteome and metabolome, respectively. With the generation of very large data sets, computational tools are needed to analyze and interpret the data. There are several types of tools available. A genome browser helps data analysts to visualize the data and to create a map of the data set, such as mapping a sequence to a specific gene. The Genome Browser by the University of California, Santa Cruz is a freely accessible browser with multiple tools. Again, with the generation of lots of DNA sequences, tools are needed to help align or match the sequences to a reference map or to other sequences if a reference map does not exist. New variations can be discovered, or the prevalence of known variations can be calculated. In addition, given the sheer size of the data sets, tools are needed to help reduce or focus the analysis on a subset of data. Genome sciences can be used in many fields of study, in medical and non-medical research. Basic genome sciences refers to research that aims to advance scientific knowledge, but is not directly applicable or immediately useful. One example is to explore the non-coding regions of the genome, which account for about 97% of the genome, to identify conserved regions between fruit fly, mouse, and human. Applied genome sciences refers to research that aims to address or answer a question about a specific application or resource for an intended purpose, such as developing a new test for cancer. One example is to identify a pattern or unique collection of genetic variants that predisposes an individual to a disease. For more information about genome sciences and current research programs, please see the National Human Genome Research Institute website of the NIH or the GCB website. This concludes this module.